Hello everyone, my name is Nguyen. Today I will present my research proposal about how to predict alcohol consumption using machine learning and telehealth. Alcohol use disorder is characterized by loss of control over excessive drinking behavior over time despite adverse consequences. During the pandemic, online sales of alcohol has doubled compared to last year. Excessive alcohol use may also lead to anxiety or depression, which themselves been increasing during COVID-19. Telehealth is now becoming the default treatment modality. However, these devices face limitations in detecting alcohol use disorder, such as assessing certain aspects of speech and psychomotor agitation. So the study aims to predict alcohol drinking behaviors from neuronal activity in a specific neural pathway. These data can then be integrated into the telehealth system, which supports detecting alcohol use disorder. Recent studies found that the inhibition of the top-down circuits from the prefrontal cortex promote compulsive drinking. Specifically, direct projection from the anterior cingulate cortex to the stratum play a key role in alcohol-seeking behavior. However, how specific features of neuronal activity in the ACC striatal pathway are affected by alcohol consumption and whether it can predict excessive drinking remains unknown. In the Huda lab, we previously examined the changes in neuronal activity of the corticostriatal neurons using 2-photon calcium imaging as head-fixed mice actively drank alcohol over days. Transgenic mice expressing GCAM6F were injected with retrobeats in the stratum to label corticostriatal cells. Free-moving mice were habituated to ethanol drinking through the drinking in the dark paradigm, followed by drinking while head-fixed. Labeled and unlabeled cells were recorded simultaneously with pupil size and leaking behaviors. In this project, I will begin this analysis with support vector-machine decoder, followed by more advanced classifiers like artificial neural nets. Specifically, I will test whether neuronal activity can be used to predict drinking versus non-drinking, level of intoxication, and concurrent motor movement. Neuronal activity from corresponding temporal session will be compared. For example, to test for drinking versus non-drinking, classifiers will be trained to distinguish activity during pre-drinking and drinking sessions. For level of intoxication, classifiers will distinguish between activity on early and late drinking. I will minimize model overfitting by using the cross-validation techniques. This way, the model is fit on data that is not used for the prediction, thereby increasing the generalizability and robustness of the model. In conclusion, this project applied machine learning to identify the relationship between neuronal activity and alcohol consumption. With the help of AI and telehealth, these findings may potentially predict alcohol use disorder from patient-generated neural activity, which plays a special role during the COVID-19 pandemic.